So Carolus Linnaeus's work in classifying and categorizing and naming organisms was a really important step in cataloging all of the diversity on Earth. But he was just using similar physical characteristics. About 100 years later, Charles Darwin came along and started proposing a mechanism for and documenting the idea that organisms could change over time and give rise to new species. And so Darwin began classifying species based on how their common ancestors and how they were related. So the modern version of this classification system is called systematics. So systematics classifies species based on their common ancestry. So Darwin used the idea of a tree with a common ancestor and then all of the related species coming off of it. And each of those nodes was the divergence of different species. So we use the idea of systematics in order to reconstruct the phylogeny or the evolutionary history of living organisms. So we classify organisms based on their common ancestry and how they are related to reconstruct this evolutionary diagram of how we think species are related to one another. A classification system based on shared traits is called cladistics. So I know this is a lot of vocabulary words. These are sort of interrelated terms, and they are all make use of the idea of shared ancestry and shared traits in order to figure out how living organisms are related to one another. Now I hope that this video is actually a short one because we're going to come back to this idea of systematics and phylogeny in our next two labs. So we're going to do labs that allow us to reconstruct evolutionary trees based on shared characteristics and common ancestry. So this is how a phylogenetic tree might look today. So if we look at a very broad phylogenetic tree of animals, we can make a hypothesis about how they're related. So each one of these nodes or separation points indicates that there was a common ancestor. So way back here, a long time ago, we had a common ancestor between fish and all other animals. Then a little bit farther down, a little closer in time, but still a long time ago, there was a common ancestor of birds, humans, rats, and mice. And we can see these common ancestors indicated all along this tree. So phylogenies are a hypothesis. They can be based on physical characteristics, they can be based on DNA and evidence, and sometimes a combination of both in order to figure out where our common ancestors were, what they might have been, and how closely related they are to one another. One thing that phylogenetic trees doesn't tell us is how the relative level of evolution of each of these organisms. So we are not more evolved because we've branched off farther along than a fish. We've simply been going down our evolutionary trajectory since a common ancestor at the same amount of time. So we've had some more splits than, say, this particular fish, but that doesn't mean that we are more advanced, or they are more primitive, or we are more evolved. All of these organisms their ancestors have been evolving for millions of years, and so they're all highly evolved and highly adapted to their specific environments. So with these trees, we can actually spin them on the nodes and place the common ancestors in living species in different orders. It doesn't matter as long as we keep intact the position of the common ancestor. So this tree on the left indicates the exact same information as the tree on the right. Humans just happen to be on the right instead of the middle. So we can spin these as long as we keep everything with respect to their common ancestors. I did a quick edit there. I just took out the little blurb at the top because it actually applied to the slide before. So this is our big evolutionary tree of life. You'll notice that it shows the three big domains: bacteria, archaea, and eukarya. And then in here we're showing the all of the eukaryotic kingdoms. So we've got the animal kingdom, the fungi kingdom, the plant kingdom, and then we also have this protist kingdom, which I'll come back to that in just a minute. So initially, phylogenetic trees were made and still can be based on physical characteristics. The problem with this is you have to choose the correct characteristics. You have to choose the ones that are actually relevant to the species and the common ancestor, and you don't always know what those are. So now that we have DNA sequencing technology and we have a lot of molecular tools to analyze different sequences, different proteins, different genes, we can use that to determine ancestor and descendant relationships as well. And so you do this by looking at differences in genome sequences between different organisms. So over here we have a blow up of the same animal kingdom or animal groups that we had on the previous slide, but basing this off of differences in DNA sequencing. So this example shows using restriction enzymes. We used restriction enzymes in one of our lab. They're little proteins that cut DNA at specific sequences. And so you can look at different enzyme restriction patterns for specific genes to look at how these organisms are related to one another. One of the things that we can look at in a phylogenetic tree is how the members are related to one another and how and where they share a common ancestor. Individuals that are grouped together by their most closely related groups and their common ancestor are called monophyletic. Phyla meaning group, mono meaning one, so one group. So all of the organisms with their common ancestor are called monophyletic. So all descendants of a single common ancestor are going to be within the monophyletic group. You can also look at organisms based on similar characteristics, but that don't have a common ancestor, or at least not a common ancestor that includes all others. So crocodiles and lizards share many physical characteristics. And if you based your genetic or your tree based on just these particular characteristics, you would end up with something called a polyphyletic group. So poly means all organisms that are descendant of a particular group but that don't include others. So if we're talking about crocodiles and lizards and excluding birds, these guys are polyphyletic because we're leaving out one of the groups that has a shared common ancestor. Now we could also go back to before B and make a monophyletic group out of birds, crocodiles, and lizards. So monophyletic means a common ancestor and all of its descendants. Polyphyletic means that there's a common ancestor, but that we're not including all descendants of our group within the group that we're discussing. So there's a couple of different ways that you can construct evolutionary trees. So you can construct them using anatomy. And the problem with this is that it doesn't necessarily, you have to think about which traits are the most important, and you have to do a lot of trial and error before you actually get to the quote-unquote correct phylogenetic tree. Now some of this is going to be based on where the organisms are located. So if we're looking at these animals in North America, just thinking about them, they're probably more closely related than they are to animals that are located in Africa. So we can use biogeography and distribution of species to help us with this. But in actually assessing the anatomical features, we can use similar features in order to group them by most similar features. So in this example here, we have the African lion, the spotted hyena, the gray wolf, and the black bear. So we can look at their different features and construct an evolutionary tree from that. Do they possess retractable claws? yes, these guys on the left do possess retractable claws. That's one indication that they might be related, whereas these guys don't. We can also look at the number of chambers in the ear bone. These guys have 2, so there's another feature they have in common. Wolves and bears have only 1, so that's another feature that they share. Does the penis have a bone? No, the African lion and spotted hyenas don't have that trait, whereas the gray wolf and the black bear do. So in considering that they share these 3 traits, and these individuals can share these 3 traits, we can separate them from a common ancestor, assuming that these differences have accumulated since that split. We can also construct an evolutionary tree using DNA. And so we do this the same way that we use comparative anatomy. You look at the differences in the DNA base pairs. Now, usually this would be a particular gene that you think is present in both, but that has changed or diverged over time. So if we look at species 1 and species 2, there are quite a few base pairs that are similar between them. In this case, we have only 2 differences. If you look at 3 and 4, they have more differences between them, but they don't have as many differences as between species 2 and 3. And so you can look at the number of differences between the different groups to determine where you think they are related. Now, if you look at the relative positions of these are indicating time. So this suggests that these 2 species have diverged less recently because less changes have accumulated in their DNA. Species 3 and 4 have diverged a bit farther back, and you would come to that conclusion based on the fact that there are more differences between 3 and 4. Now between species 2 and 3, you have a lot of differences. This indicates that their common ancestor that shared a common DNA sequence happened much farther back. So sometimes we have little hiccups when we're attempting to reconstruct our phylogenetic tree. If you remember from chapter 10, we talked about homologous and analogous structures. So remember that homologous structures are structures that are similar because of a common ancestor. Analogous structures are structures that are similar due to convergent evolution, meaning they're structures that come about due to an adaptation in similar lifestyles or environments. So if we are looking at a bat and an insect, at first glance we might say, oh, they must be closely related because they have wings. But when you start looking at other features of common ancestors, you realize that bats have more things in common with humans who are also mammals than they do with insects. Now that would suggest that this trait of wings evolved separately in the bat and the insect line. They evolved because of specific environmental pressures and occurred way after split from common ancestors. And you can see that the bat had a split much, much later. Homologous structures are what we're going to use in general to construct phylogenetic trees, but analogous structures can cause some problems. So let's look at another example of convergent evolution. So if you were looking at this little shrew and this African golden mole, if you were looking just at physical characteristics, you might put them in the same group. They live in similar habitats, they have fur, they have similarly shaped noses, but when you actually look at DNA evidence, it allows you to sort of separate what is an analogous trait and what are homologous DNA features. And it turns out that even though the common shrew and the African golden mole look more similar and might be placed together in a phylogenetic tree based on physical characteristics, when you actually look at DNA sequences, you find that the African golden mole is more closely related to an elephant. Now, that doesn't mean that their ancestor was yesterday. It just means that the common shrew and the golden mole have adapted to similar environments, but that there was a similar ancestor, a common ancestor, between the African golden mole and the African elephant, and we can deduce that from DNA sequence comparison.